Good afternoon, ladies and gentlemen. It's a great pleasure to have you in this concurrent session. If you're here and you are hoping to see crop performance and nutritional quality, you are in the right spot. I realize we're a little bit distributed because I realize also the screens are at both sides of the room, so we will be comfortable with that. My name is Stan Blade. I'm Dean of the Faculty of Agricultural, Life, and Environmental Sciences at the University of Alberta. We have about 1,600 undergraduates, about 530 graduate students, and we have, over the last 5 years, an annual budget of external funding of about $45 million, very active in production agriculture, but other aspects of the environment, of food, of nutrition, and it's a great pleasure to have you all here. We have a tremendous session. Credit to Morris and the team. It's just been, I had high expectations coming to Saskatoon. They have all been met and more. And the speakers that we have in this session, again, I'm sure will meet that high bar. So credit to the team that has brought all of this together. I'm going to introduce the first speaker. In so many years of chairing international sessions, I've never been able to use this line before. I am responsible for knocking out the tooth of the daughter of our first speaker. You know, back in my days with IITA, Lena's daughter came over to a birthday party of one of our children, and there was a very active balloon volleyball game, and mistakes were made. Let's just say that at this stage. So our first speaker is indeed Dr. Lena Trapathy. Lena is a senior scientist with the International Institute of Tropical Agriculture. She leads the plant transformation unit. You will hear a remarkable story this afternoon about her work with banana. And I would only identify just one element of her biography, that she has trained 30 graduate students and 200 short-term trainees, many of those from sub-Saharan Africa. Please join me in welcoming Dr. Lena Trapathy.
Good afternoon, everyone. First of all, I would like to thank the organizer for giving me this opportunity to talk something about my work. So what I'm going to do today is I'll talk particularly about the bananas, the GM bananas resistant for the Xanthomonas wilt and the nematode resistance. And why bananas? You know, banana is like a fruit crop all over the world. But in Africa, it's a staple food crop and is the fourth most important crop. The overall production of banana is about 144 million tons per annum. And 1/3 of that comes from Africa. Particularly Uganda is a big grower, is the second largest producer after India. You know, but in terms of the consumption, actually, Uganda is #1. And they can eat bananas, but they don't eat like a fruit. They have a staple food called matoke, which they can eat starting from the breakfast, lunch, and dinner. So you can understand how important that crop is for them. But still the production of banana is threatened with several bacterial, viral, fungal diseases, but as well as a lot of pests like weevils and nematodes. So at IITA, we are collaborating with a lot of partners. We are focusing on pests and diseases. But today, keeping the time in mind, I will give you two examples. One is the Xanthomonas wilt and the nematodes. Those are the two projects where we are actually much more advanced in comparison to the other projects. But we are also tackling the viral disease called banana bunchy top virus, which is also a big problem in bananas. So Xanthomonas wilt is caused by Xanthomonas campestris pathovirus mysosurum. It's not a new disease. Actually, it was reported in Ethiopia more than 45 years back on crop called enset, which is similar to bananas, but is not bananas. And then after that, it was confined in Ethiopia for a long period of time. Then in Uganda, it was reported in 2001. Since then, actually, the disease is spreading very fast, and now it's covering all over the East and Central Africa, the Great Lakes region, which is the major banana producing area in East Africa. And so it's actually in DR Congo, Rwanda, Burundi, Tanzania, and as well as in Kenya. So the pathogen is very closely related to a bacterial, which is Xanthomonas vesicola pathovirus vasculorum, which is a pathogen for sorghum, for sugarcane, and it can also affect maize. So the Xanthomonas for banana, Xanthomonas pathovar actually is very, very closely related to that. For a long time, actually, everybody was thinking that, is a monomorphic pathogen. But recently, there was some sequencing, genome-wide sequencing was done in a lab in UK, and they revealed that, no, it's not monomorphic. Actually, there are two different groups. So Uganda, Kenya, and Burundi is in one group. But then DR Congo, Rwanda, and Ethiopia is actually in a different group. But still, the pathogenicity of this one is quite similar. The production losses are pretty high because once the disease enters into the field, actually it's completely wipe off the bananas very quickly. So the production losses are up to 83% in DR Congo and up to like around 70% in Uganda. So you can see that much loss of bananas are really huge. And as I said, because it's quite extreme and rapid, so within few months, once the disease is in the field, it can wipe off. And you cannot miss the disease once the disease is there, because it starts with the wilting of the leaves, the premature ripening of fruits. So even the fruits which looks quite good from outside, when you cut them, they are all rotten from the inside. They have this brown scar. But also there is a yellow ooze, which is like a peculiar symptom, and you can easily diagnose the disease because of that. The disease mainly spreads by insect vectors. So what happens, in banana, there is this flower. In the male flower, the one, like a bract falls off, and it leaves the moist cushion. That's attract the insects. And that's how the insects carry the pathogen from the infected bananas to the non-infected. That's the major spread. But then it also spread through the planting material, because I don't know how many of you know that, banana, there is no seed. So it's actually planted using the suckers, which are the babies coming out around the bananas. And then if the mother plant is infected, then those suckers can spread easily the disease. The farmer spread it also using the contaminated farming tools, and also through the trading, because they use the banana sheath and the pseudostem sheath to wrap the bananas, and that's how the pathogen also spread actually long distance. In terms of the susceptibility, actually all the banana varieties so far tested have been found susceptible. But then there is not much significant difference in the pathogenicity among the different isolates. There are several labs who has done that work, including my lab. So we have tested 50 different isolates collected from different countries, and we didn't find any difference in the pathogenicity of those ones. Even though all the cultivars are susceptible, but there is difference in the susceptibility among the cultivars. Like Pisangawak, which is a beer banana, is the most susceptible. And the interesting thing is there are some cultivars of particularly the East African highland bananas like Nekitembe, where they can escape the insect-mediated transmission of the pathogen, because they have this persistent bract, so the bracts doesn't fall off, but they are not resistant. So if the farmers use the contaminated farming tool, they still get the infected. There is only one wild type banana, which is Musa balbisiana, which is found to be resistant. But then, this is not the preferred parent for conventional breeding because of other problem. Like bee genome has this integrated banana streak virus problem, so breeders don't use it. And even though there is a resistant cultivar, banana breeding is a very complicated, difficult thing. So the disease can be managed using the cultural practices. But then there is also, the cultural practices are not well adapted by the farmers because they're very laborious. But also the farmers themselves are very confused, to just uproot the main plant which is infected, but also whether to uproot the whole mat. So, one of my master's students, we were very curious to see what's happening. So we did this work with the master's student, and the student did the work. So what she did was she infected the male flower, which is the natural way of doing it. And then we used the PCR to detect the pathogen. So before she can detect, before they can detect the symptoms, you can detect the pathogen actually even in the sucker. So you can see it's a vascular pathogen, so it moves very fast. So, like recommending farmers only to uproot the plant which is infected is actually quite risky. So I think I'm now pretty much sure. I was trying to justify why we are doing the transgenic research, because we, as Jennifer said yesterday, is not a silver bullet. We need a strong justification because it's a quite expensive and as well as the lengthy process, So there are overall economic losses between 2 to 8 billion over a decade. And as I said, it leads to the absolute loss. So that's why, even for the other bacterial diseases, resistance has been considered as the most cost-effective, and there is no resistant variety, so conventional breeding is not possible. That's why we started the project on the transgenic. So what we are doing is we are using 2 different genes. One is plant ferredoxin-like protein, and another is the hypersensitive assisting protein. Both of them are cloned from the sweet pepper, which we all eat, that green bell pepper, the capsicum annum. And what these genes do is they intensify the hypersensitive response, and it is actually a harpine elicitor-induced resistance, so which can basically provide the broad spectrum, and that's why we are trying to use it. And this gene has been tested in several vegetable crops before, and there has been shown resistance against different bacteria, including even Xanthomonas. It has been tested in rice before, but nothing has been tested under the field condition. Everything was done in the glass house and the laboratory condition. So what we did, because our target was not like an upstream research only to publish, we were always thinking of providing the solution to the farmer. So from the very first day, we developed the project considering all the aspects of the GM product, including like IP, including how we are going to disseminate the product once we are ready with the product. So what we did was we partnered with AATF, so the technology we got was that both the genes are actually from Academia Sinica, Taiwan. And one of these genes they have the patented it, not even in Africa, but all other places. So what we did was IATA approached AATF, and AATF negotiated with Academia Sinica for royalty-free access to the gene. And so now we have actually freedom to operate, and we can actually go up to the commercialization without making any payment. So the advantage of that is like farmers doesn't need to pay anything extra once the product is ready. And then this project also has a lot of capacity building component because we are working very closely with the national partner. So we started it by developing the capacity for the transformation. Transforming banana is not easy. And then we went to all the steps actually we were doing the capacity building, including the glasshouse testing and the field testing. And in the end, we have the public-private partnership. So we are also actually involving a lot of tissue culture companies from the starting itself so that they have the capacity to disseminate the material once our product is ready. So we developed, so it was a bit easy for us to go on the product because, we had the very robust banana transformation platform using the cell suspension. Developing cell suspension is quite tricky. It can take 18 to 24 months. But the good thing is once you have the cell suspension, you can use for the transformation for at least 2 years. You have to maintain the cells. And then once the cells are there, it takes between 9 to 10 months actually to develop the transgenic bananas. So that's what we used. We have actually, in the starting, for the proof of concept, we transformed 2 varieties. One is Succulentese, which is a desert bananas grown in East Africa, is a small apple bananas. And another is an Echinacea, which is a East African highland bananas. And we developed actually about 100 lines for each cultivar using each of the construct. We use constitutive promoter for driving the genes because, as you can see, the pathogen can be affected from anywhere in the plant, so we cannot use the promoter for tissue specific. And then after developing the plants, we validated for the presence of the gene, and after that, we went for the testing. So what we did was we developed a quick screening method using the in vitro plants where we can inject a very small in vitro transgenic plant. And the good thing about this is, like, between 10 to 12 days, the control plants gets the symptom. So it's really quick, but we keep them at least for 60 days. The plants which don't see the symptom in 60 days, we take them out for the glasshouse study. And in the glasshouse, actually, we found, because we have already done the preliminary screening, so most of the lines actually showed resistance when we went to the glasshouse. You can see that blue tag is the one where we have inoculated the pathogen into the leaves. The control plant actually got the symptom within 1 month to 40 days time. They have completely wilted. That's in figure C in that picture. And then the control, and then the transgenic plants, actually, majority of them didn't show any symptom. We also tried to see, what's happening. So what we saw is, the point where we have inoculated the pathogen, at that particular point, you see the necrosis. That necrosis is because of this cell death, which creates the physical barrier, and that's how normally the hypersensitive response is, so it creates a barrier. So I wanted to see, whether exactly that thing is happening. So what we did is, we tried to isolate the bacteria 5 centimeter above the point of inoculation and 5 centimeter below the point of inoculation. We couldn't get any bacteria, which was proving that, means our hypothesis was correct. And we were not only doing it by one way, by PCR, but also on the selective media. So we tested by 2 different performance. Then we have, based on the glasshouse study, we selected the promising lines, and we did the detailed molecular characterization for the copy number, for the expression, everything. And based on the molecular characterization and the glasshouse study, we selected 65 independent events. 45 of them has the HRAP gene, and 20 of them has the PFLP gene, and we planted them in the confined field trial in Uganda. All this research, actually, we are doing in collaboration with NARO in Uganda. So we planted that, and then our target was to do the disease evaluation for BXW, but also to do the agronomic performance, to see whether they have the similar yield similar to the non-transgenic, because if the yield is affected, we are actually going nowhere. So what we did, this disease is actually spread through the insect, but when we got the approval, we were supposed to remove the flowers. We were supposed to bag the bananas until the flowers are removed. So that way, we have to do artificial inoculation, because this is not a soil-borne pathogen. It normally stays only for few weeks, can survive in the soil, after you remove the bananas. So we have, again, artificially inoculated in the leaves. We use the pre-flowering stage of the plants, and we inoculated them. And after that, actually, the picture was quite black and white. Several of you might have seen those bananas in the field. So, we don't need to explain to them which one are the control plants, which are the transgenic bananas and all, because the control plants got the symptom. Most of them didn't produce any fruits. Even though the fruits were produced, they were all symptomatic. Majority of the transgenic bananas actually didn't get any symptom. But that was obvious, because they came out from after screening in the glass house. So we tested these plants for 3 generations, the mother plant and the 2 ratoon crop. We inoculated the first time the mother plant, but the second time we inoculated actually also the first ratoon, but then after that we didn't inoculate it, because there was what we observed that between the connection between the mother and the ratoon plant, that was giving the pathogen to the ratoon already. And as you can see, that hole in the control, that there was the plants there, but as soon as these suckers were coming up, they were all dying off, because there was enough pathogen there. So these are the pictures of the transgenic and the non-transgenic bananas. As I said, control plants, majority of plants didn't produce any bananas, because we inoculated them at the pre-flowering stage and they all died. But even though the bananas, the bunches were produced, they were all symptomatic. So what we also did was, We also checked for the yellow ooze. The lower panel is the non-transgenic banana, and the upper panel is the transgenic banana. So we didn't find any pathogen or any ooze in the promising lines of the transgenic bananas. So we published this in Nature Biotech. And so what we found is the result actually for the 3 generation was quite surprising. You know, there were 11 lines which has resistance in the mother plant as well as all the generations. So that was like a stable trait. We find some additional like 5, 6 lines which have like, you know, only the leaf which has been inoculated got the symptom, no symptom, further spread of the symptom. But when it come to the ratoon crop, there was no symptom at all. So we had those type of lines. We have also some of the lines where like, but those were very few plants that where there was no symptom in the mother plant, but there were symptoms in the progenies. But we have 11 lines were good enough for us to move further. And then, as we said, we were checking the plant morphology. So these lines, the good lines, has no significant difference. They look normal, and also the yield parameters. So for the morphology, actually, we were not only by the look, we were actually measuring. We were counting the number of days to flower, the height of the plant, actually the leaf area, and the pseudostem girth, and there was no significant difference. For the yield also, we weighed the bunches. This was bunch weight, the number of fingers, and there was no single. So, you know, like I said, that we were using 2 different varieties. So we compared actually the transgenic of one variety with the non-transgenic of the similar variety. And the point which I would like to make here is, for the yield, actually we compared the inoculated transgenic bananas with the non-inoculated control bananas. Because, you know, that the inoculated control bananas died, you know, so that was the comparison. And then after that, I was quite curious to know like what's happening, why some of the bananas has the partial resistance, some has the full resistance. So we wanted to see whether there is a difference in the expression. So we did the qRT-PCR. We picked some lines which was showing the partial resistance and the lines with the 100%, but we find no correlation in them. So, you know, which was not surprising, because most of these hypersensitive response genes, you really need a small expression which is enough to provide the full resistance, and that's what we confirm from there. Because this was the first trial for me, so I was also, for the curiosity, I also wanted to see, like, you know, the stability of the presence of the gene and the expression of the gene, you know, what's happening. So every 6 months, actually, we were doing the PCR and the RT-PCR on these plants, and we actually found that the trait was quite stable and the genes were stably present there for 3 years. After that, we went back to the regulators. We said we want to do the second trial, and they gave us the approval for 10 lines. So we did the second trial with 10 lines with more replicates, and the result was the same. This time, all the 10 lines retained the resistance, 100% resistance. We tested them for 2 more years. So far, actually, we have the result for 5 years in the field with 5 generations, and then the trait is quite stable, and we are actually now moving to the product development. So we have proof of concept where we have done the gene delivery establishment. I think we have actually put the baseline for everything. And now we are more into the variety. We are not developing one because farmers doesn't grow one variety. We are actually developing 5 varieties. We have recently got the approval for multi-location trial in Uganda for starting the trial in 3 different locations. We are also waiting for the approval from Kenya for going for the field trial in Kenya as well. So, but another thing what we are doing is, you know, the single gene-based resistance is risky. So we are now stacking the genes together. So now whatever we are going to the field actually is with the stacked genes together. We have developed the lines for all the 5 varieties. We have tested them in the glass house, identified the best lines to go into the field, and like in a month or 2 months' time, actually, we will be planting the trials both in Uganda and Kenya. And if everything goes well, our plan is by 2020 or 2021, actually, the product will be ready for deregulation in Uganda and Kenya. Maybe by that time, the law will be in place in Uganda. In Kenya, there is already a law. So we have done the biosafety analysis using the bioinformatics, and there is actually no convincing evidence to say that, you know, there is any alarming thing there. Initially, I did it myself, but then, you know, to get it regulators' confidence, we also did through the third party. So Richard Goodman did that for us. The good thing is, like, a team of the socioeconomists from IITA did the ex-ante impact analysis, and I was very happy to see that there are 65% farmers who says that, you know, they will adopt these bananas immediately as soon as they are ready. There are 19% which says that, okay, they will do it later. After that, they see how they are performing in their neighbor's field. There are only 16% who says that, you know, they will never, they might change their mind as well. And then, so as I said, you know, this disease was reported on NSAID, so Ethiopia still has a problem on NSAID with this. So we were approached by Gates Foundation that if we can help Ethiopians for transferring this technology to NSAID, and that's what we are working on with EIR in Ethiopia, and we are transferring these two genes into NSAID. My next story is, which I will go very quickly, is on nematodes. And I know, like yesterday, somebody was saying there are genes. Yes, I agree, there are genes, you know, but there is not one nematode in the soil. There are a lot of nematodes at one point, at one place, you know. So in bananas, there are gene, but they are particularly provide resistance only to Rhodophilus. But Rhodophilus is not the only one in the soil. So we are working, and here IIT is working with University of Leeds in UK, and we are trying to actually develop the broad spectrum resistance for the nematodes using two different technology. One is the synthetic repellent, this is a peptide, and also another one is an antifeedant, which is a cystatin. We did the same thing, developed the transgenic bananas, and so in this case, we used plantain. We tested them with the molecular analysis, did the glasshouse assay, and then identified the best lines and did the trial. Again in Uganda. So we planted 12 different lines in Uganda with a transgenic plantain, and the result was good there as well, you know. So we used three different things like peptide alone, cystatin alone, and the stacked ones. And the interesting result here is actually we found better result where we had the peptide alone in comparison to when we have stacked these two genes. We have a few lines which has actually more than 90% resistance under the field condition. So right now, what we are trying to do is, so initially we use the constitutive promoter, but for the nematode, you don't need to use the constitutive promoter. So we are actually, we tested some root cap specific promoter, which we have also recently published, and there is a maize root cap promoter, which can actually, you can express the gene only at the tip of the root, and that's enough to actually express the repellent. So right now, we are planning to use this promoter to drive a repellent and then go for the product development from this. So in conclusion, I would like to say that GM approaches shows enormous potential. And I think the point which didn't came out yesterday was one trait is good, but you know, stacking the different traits, you know, like in bananas, there are 5 or 6 different problems. So if we can make a super banana by stacking all these 5, 6 traits together, that's our goal. So we already started working on some of those traits together. I didn't have the time to actually talk about that. There are several products on horizon for Africa, but BXW resistant banana is one of them, and our target is actually to deregulate that by 2021. So in the end, I would like to acknowledge all my partners from the national program, AATF, Academia Sinica, and also the financial support from different donors. And then last but not least, my team working with me. Thank you very much.
We have time for some questions for Dr. Trapathy, if you wish.
Thanks, Lena. I'm one of the people who's had the benefit of actually seeing your transgenic
Yeah.
bananas in the field at Narrow. What's interesting is how difficult the political situation is in Uganda, and you're very familiar with this, of course. What I don't understand is how it's possible for the field trials to be approved. So there is a regulatory process for considering genetically modified crops, and yet all of your colleagues seem to be of the opinion that there must be a biosafety law passed in order for there to be, wide commercialization. Now, why are you able to proceed with field trials, but you consider it's not possible to proceed with commercialization? Why not proceed with commercialization even under the current regulatory system?
Yeah, so actually Uganda has the biosafety guideline in place, and the guidelines allows the confined field trial. So right now the open field trials are not allowed there. So like you can go multi-location trial, but they all need to be confined. So it's expensive, but you can do your research. But for the deregulation and commercialization, you need the law in place, which is right now in Uganda is not there. I hope I have answered your thing.
Time for one quick question, if anybody has one. Hearing none, let's thank Dr. Trapathy. Morris, she just come all the way from Nairobi. We're going to get her a full set of questions. Morris.
Yeah.
So I just wanted to clarify, Lena, what the mode of action of the ferredoxin-like gene that you're using for Xanthomonas wilt resistance. How does it actually function against the bacterium?
So actually it activates this oxygen burst.
Okay.
Yeah, so that's how, actually it's a defense
Right.
gene.
Yep.
And that's how it works. So, it has the oxidative burst, which then leads to the systemic acquired resistance.
Okay. So
That's
it's
how-
ROS
Yes,
mechanism.
it is. Yeah,
okay.
AOS method, yeah.
Super. Let's thank Lena once again.
Yeah. Okay, thanks very much.
Our second speaker this afternoon in our session is Dr. Sigrid Hoyer. She is the science coordinator and program leader for nutrient-efficient crops at the Australian Centre for Plant Functional Genomics. Dr. Hoyer started her work on Nerica rice with the Africa Rice Centre. Then, as you heard yesterday, did significant work at Erie for a number of years before moving on to Australia. Dr. Hoyer.
Okay, thank you very much for the introduction, and thanks, Maurice, for having me. It's my first time in Canada, and I'm enjoying it a lot. Also, the talk so far, very exciting things going on. So hard to beat my predecessor here, but I hope you enjoy also what I have to tell you. I'm going to talk about wheat, which I started working on in Australia, but also about rice. Okay, is that better? Yeah. Okay, so this is a rather old slide that I like to show. This was prepared in Erie, and it's showing you the increasing demand and the increase in rice productivity we have to achieve to keep it pace with the increasing demand. And the red bars showing the increase in Asia and the blue bars in Africa. And this is based on data from 2010. So we're preparing for this talk. I thought it's maybe a good idea to update that. And I was quite pleased to see in the FAO database that the global rice production actually is 488 million tons and 494 in 2015. So it's actually exceeding the required increase in productivity. And I like to think that this is probably due to all the efforts going on in rice breeding, so it's actually paying off, and investment in agricultural research is paying off as well. So we need to make sure that this stays this way. So when we look at rice, there are two main production systems, the irrigated, which is generally favorable, and the stress-prone rain-fed environment. And the irrigated system is more productive, so 70% of the rice is produced on about 60% of the area. And this is due because we have constant water control, and with that also some control over wheat. And because it's a safe environment or low-risk environment, farmers also apply fertilizers. In contrast, the rain-fed environment is 40% of the total area in Asia, but only 25% of the production. Obviously, this is due to drought and floods, as I will show you later. But because it's a high-risk environment, also farmers are reluctant to put a lot of inputs, fertilizer or other agrochemicals. It's just because the risk to lose the crop is too high. Also, they don't have the means or the access to fertilizers often. So if we want to make progress in those systems, we really do have to develop stress-tolerant varieties. And at the top of the list is drought stress, but at the same time, because we are in a monsoon region, we're also dealing with submergence stress. Nutrient deficiencies is an issue, but also toxicities, aluminum and iron, which then in the end also causes P fixation and P deficiency, heat, salinity, you name it. So there's a lot of scope to improve productivity in those systems if we have stress tolerance. So the first project I want to talk about is submergence-tolerant rice. Most of you are probably familiar with this project. I don't want to take all the credit, so Dave McKill was the one who started this and also really brought this to the success. And Julia Bailey-Saris in UC Riverside, she did a lot of the science, the molecular and physiological study on this project. So what is the issue when fields get submerged, so completely submerged? The plants try to escape. So they show this leaf elongation, they go to the water surface. And in deep water rice systems, the water stays and the grains can be harvested from boats. But when in submergent-prone environments, then the water at some point recedes, can be several meters of waters for a few weeks, and then the plants lodge, and actually that's the end of it. So what we have identified at the time, or Dave initially, is a major QTL on chromosome 9 that confers tolerance. Oh, sorry, that was too early. And we had the donor variety of this trait is FR13A. It's a landrace, an Aus-type variety, and we sequenced the QTL region, and you see on the top the QTL region in the Tolerant variety, and then in comparison, the Nippon-Bari reference genome at the bottom. And what we find was we did find 2 ERF transcription factor genes, so ethylene-responsive genes, perfect candidate genes, because we know submergence is related to ethylene response. But then when we had a closer look, we found there was a 3rd gene present in only the Tolerant variety, which is absent from Nippon-Bari, and that's the sub-1A gene. And this gene, when you have it present, either in transgenics or in this case, introgression lines having the entire QTL, it's actually preventing the escape response. So it's conferring a quiescent status to the plant. The plant is just sitting, waiting until the water recedes. So it's not trying to escape, it's not growing. It retains its carbohydrate reserves for recovery growth after the water recedes. The other plants, as you can see, hugely elongated, and they don't recover. So this is a field demonstration plot in Erie. Those plants had been submerged for 17 days under several meters of water, and then were allowed to recover. And you can see here, if you compare Summer Masori to your right, and the introgress, the sub-1 QTL, you can see that the survival rate is much higher. Similar, if you put this QTL into IS64, we have a much higher survival rate. So this is a really beautiful project, and it shows 2 things at this stage. So there's a single gene that can actually have this major effect. Previous to that, submergence tolerance was considered as a very complex trait, so many QTLs involved, and now we know it's just a single gene that can do that. And the other point I want to make here, it's absolutely essential to put those kind of genes or proof of concept into different genetic backgrounds, and we've heard this before, because otherwise there's no adoption, and certainly not in rice, where you have very regional consumer preferences. So this Swana sub-1, Swana is an Indian variety that is very well widely grown, and that was the first sub-1 variety that actually made it into farmers' fields. You can see here, after this field was flooded for several days and repeatedly, there was, in the end, the plants recovered and we had a happy farmer. So this is really beautiful to see this working in the field. Swana sub-1 was tested across many sites, and they had about 1% on average yield advantage over the controls. The third point I want to make with this project is that to really get it out into farmers' fields required a lot of resources, a lot of time, and a lot of money. And Bill Gates at the time came to the rescue, and they pushed IRRI to do the seed production. IRRI, until then, actually didn't do that because they thought it's in the national systems who should be doing that, and then handed like pre-breeding products or lines to the national system. So that was the first time IRRI did this, and Umesh, to the right, he did a fantastic job mobilizing all the seed producers and get it out there to the farmers. Without that, I think it wouldn't have been the success it is now. The other project I want to talk about is phosphorus deficiency tolerance, and I don't need to tell you that we need to have nitrogen, phosphorus, and partly also potassium applied to retain soil health and to have a high productive system. Again, this project has been started by Matthias Visuva. He's in Japan, and Jirkas was at some point at IRRI where we met. Chin is the one who did all the breeding marker development, and then Rico is the one who cloned the gene in the end. So this project started with a screening of a small panel of only like 30 different rice genotypes in a highly P-deficient field in Japan. And Kasalath was identified as a variety that had a much higher P uptake under those conditions compared to, again, Nippon Bari. And then Matthias went ahead to develop a mapping population, so you can see the neosogenic lines C443 in the field. They are productive, they have much higher tiller compared to the Nippon Bari. And there were two Qtls identified at the time on chromosome 6, an intermediate effect QTL, and the main effect QTL was on chromosome 12. Again, we went ahead sequencing across the region, and this time Kasalath is at the bottom, the Tolerant one, and Nippon Bari on the top. And you can see again there are huge differences between those two genotypes. And the India region is actually 100,000 base pairs which are just missing in the reference genome. And we had a very messy set of 69 pseudogenes or candidate genes, so I spent a lot of night shifts trying to figure out what is actually a gene and what is not. Eventually, we settled on two main candidate genes. I'm not going to talk about the dirigent gene because this is actually downstream of the protein kinase. And this protein kinase is the one that we named phosphorus starvation tolerance 1 or PISL 1. And I just want to mention again that this gene is in the indel, so it's in a unique region specific to the tolerant Kasalath. When we put it in a constitutive promoter into rice, we screened that in P-deficient soil, and you can see the transgenic plants have a much greater productivity, high yield, and higher tiller number. And if you compare the roots, you can see this enormous root growth of the transgenics compared to the null controls. And because it's a constitutive promoter, we thought we can as well try it in hydroponics. So that's what we did. And even under plus P conditions in hydroponics, this gene is enhancing root growth. So rather than tapping into gene starvation responses, what this gene does is really enhancing nodal root growth or maintaining root growth also under stress. We did a lot of work with markers. We also provided training to national breeding systems to make sure this is getting out there, and the breeding programs can check if they have the gene already or identify suitable donors. And we did screen a set of IRRI genotypes. And what we found was, I cannot really point it, but these are the genotypes, and then horizontally, these are the different markers across the region. And the dark color indicates this is the tolerant allele present, and the lighter colors are the intolerant allele. And we found that actually breeders have selected for this QTL already because it's mainly present in the rain-fed rice and in the drought-tolerant genotypes we looked at. It's mainly absent from irrigated rice, I think just because it's not really needed in this type of rice. So at IRRI now, PAP1 is being permitted with other QTLs, and then Matthias at Jirkas, he's still working on this. He worked on this gene in African rice, so we have a different allele in Oryza glaberrima, but it's there as well, so further indicating that this is really an important gene. And field testing of pub-1 breeding lines is still ongoing. Those of you who work on phosphorus may know it's not a trivial thing to really quantify and have statistically significant data to really show the effect. But I think overall this project is, has identified a gene that is really relevant, and the data show breeders already use it actually. The last project and rice that I want to mention is on heat tolerance. This is the variety we use for that is N22. And the graph shows you that the main sensitive stage for heat is during anthesis, so it's affecting pollen development, but also pollen shedding. And I don't want to go into detail, just mention we have worked with Milano University and also with Max Planck to do RNA-seq, and RNA-seq in combination with metabolite studies. And we have now a really nice set of candidate genes that I would really like to see validated, but we don't at the moment have much of a project, and we need to see where this is all going. All those three genes, SUB1A, PISTL1, and also N22, but also genes for salt tolerance, are all coming from one particular group of rice. And this is called the Aus type rice. So this is a SNP marker survey of the different rice genotypes we have. And the Aus type is closely related to indica rice. And the reason why we find all those valuable genes in this group of rice, and I think there are many more really hidden, it's a real treasure chest, I think, is because this land rices have evolved in problem soil regions. So this map shows the yellow and green dots indicate problem soils. And those varieties are still grown in northern India and in Bangladesh as well. So they're easily crossable. They have a relatively decent agronomic performance. They're actually really good donor materials for those kind of projects. How am I going with my time?
Well, you've got about 10 minutes.
Oh, that's good. Okay. So then can I go back? So just to say, when we have access to 3,000 rice genomes, genome sequences, I think this is a tremendous progress. And then if you can tap into those complex regions and just on the computer pull out the genes that are specific to those kind of regions, that will be just tremendously, so much faster also to get this work done. The problem maybe is that there's a lot of work being done on SNPs. SNP markers rely on genes that are present in all genotypes or most, and probably we're going to miss out on those unique regions a little bit. So that's my concern, but I think this can be fixed. So moving into wheat, this was quite a bit of a shock for me coming from rice, and then realizing that when I started, there was no reference genome for wheat. And look at the size, it's 17,000 megabases compared to 430 in rice. So that's a real shocker. But I think, there's progress there as well, and we have RNA-seq data and survey sequences. So we have resources. We need really good bioinformaticians to help us to pull out genes, but it's moving forward there as well. Wheat in Australia is growing in a stress environment, very similar to what we see in the rice rainfed environment. So the yield is really low, it's 2.5 tons on average compared to 8 tons in Europe. And the main reason for that is because it's water limited. It's always water limited. And in years where we have severe droughts, those are marked with a star, we lose another 50% of this already low yield. But the grain quality is really good, so that kind of compensates it to some extent. Other stresses that are important are heat, same as in rice as well, salt, pH issues, and soil issues, aluminium, but also boron toxicity. And I'm going to talk about some of those. So this is now work mainly being done at the Australian Centre for Plant Functional Genomics. Peter Lang, which was the CEO of this institute for many years, and there's been a lot of work being done on identifying drought-tolerant Qtls. And the variety, there's mainly, or the genotype mainly used for that is RSC875. This is the one in the middle. It's a really highly drought-tolerant genotype, and a lot of Qtl have been identified. And what I'm showing you in the graphic here is summarizing a huge amount of work where all those Qtls have been tested in different environments. And as I said earlier, it's the absolute prerequisite for breeding companies to be interested is that we can show them there's an effect in different environments. And the left side, so Qtl 1 to 5, you can see it consistently shows a positive effect, whereas the other Qtls are kind of variable in different years and different sites. And part of the reason for that is you have to be very specific, and certainly with drought, there's no one drought. So you need to know what kind of, what the problem is. And in Australia, surprisingly, when I came, I learned that for drought tolerance, you need shallow roots. And I thought, no, this is really wrong. But it's actually, if you are in a shallow, sandy soil, and you have in-season rainfall, you have to capture all the water before it is, washed away. In other areas, same in Australia or in the tropics, maybe you have deep clay soil, so that's when you need the deeper roots to access remaining soil moisture. Maybe then you don't have rainfall during the season. So to make a progress in drought, number one thing, you need to know what is it you actually need. The center has also traditionally worked in GM drought genes. This is mainly done in collaboration with Pioneer DuPont. And we are still, this is still work in progress. I don't want to talk about this too much, but we are field testing those. Again, this is, we know this, what is hugely expensive and difficult to do. We recently got a significant grant from the Australian Research Council to continue working on this Qtl, amongst other things. And we partner now with 3 Australian breeding companies, AGT, Longreach, and Intergrain, and hoping they help us to validate this, this material further. We also develop genetic populations to work with them. Nitrogen is part of it as well, but I'm just not going to talk about all this. If you want to learn more about this, I'm happy to talk about it. Now I'm running out of time. 5 minutes. Okay, so salt. Salt in Australia is an issue. So you can see here that it's affecting 1 and 2 farms. This map is showing you soil constraints in Australia, and then if you overlay this with a wheat belt, you can see that there are real issues there. This work is at the moment led by Stuart Roy, but it has been, it's a long-term project, and Mark Tester probably is going to tell you more about this. So we find genetic diversity. So this is increasing salt levels, and then to the right you find varieties with increasing salt tolerance. So mapping of this Qtl on material development is ongoing. But we do have already some quite interesting genes, and I like this because this is one of the few genes from our Arabidopsis that really seems to be working. So when you put this gene into barley and you screen it in the field, under very high soil conditions, you see it's actually conferring tolerance. Similarly, we have another gene, ABP1. This is a gene has been identified by Roberto Gaxiolo. It's also conferring tolerance to salt. It's a vacuolar pyrophosphatase, but it's actually one of the genes I find very promising because it enhances yield under control condition. It also works under P deficiency. So this is really a very interesting gene, and we are very pleased to have an IWIP project where we put the Pistil 1 and ABP1 in wheat, and we pyramid those, and we have a third gene, the NAS gene, that enhances zinc and iron in the grain. So this is, has been funded this year, and I look forward to working on this project. The last trait I want to mention here is boron toxicity. This is also one of the really long-term projects of the ACPFG and others, other collaborators. And this is a tolerance gene that was identified in barley. So you can see here that Sahara and the high boron conditions is not showing those leaf bronzing and symptoms. And the trait is due to a duplication of a boron efflux transporter, BOT1, and you can see in the tolerant variety we have 4 copies of this gene as opposed to 1 in the intolerant one. And then in wheat, again, genetic diversity is there. So we have Holbert as the tolerant donor of this gene. The gene was cloned by Tim and his team, And the top one is actually the functional gene. You see it's a very complex gene, and in sequencing across other genotypes showed that this is the truncation insertions in the promoter. All kinds of things happen to make this gene non-functional. A marker survey then showed that in the southern part of Australia, you actually have boron toxicity. So breeders, again, they have selected, so the red one is the functional gene, selected for this gene for the southern region, but have selected against it in the north, because in that region you have boron deficiency. And that's one of the other issues I want to say, because breeders really are reluctant to use this now, because they cannot afford to have a disadvantage of a gene in any certain region. So one requirement really is that you have genes that have no disadvantage under normal conditions, and also no yield penalty. So beautiful project, but probably not going to get very far. This is my second last slide. I just tried to, this is really a really rough one, and I need to think a little more about it. But what I'm trying to do is just to show you the pipeline. So if you do a forward genetics approach, you start off with identifying a tolerant genotype. You identify the QTL, you find MABIP, develop the markers, and then you need to validate it. So this already is a very optimistic schedule there. So it's actually rather twice as long. So the point I want to make is already after 3 years, usually funding is ending after 3 years, you just get stuck with the QTLs. And that's why there are so many papers out there, so many QTLs that never get anywhere, because funding is just ceasing. So at some point then, the more reference genomes we have, the faster this will be. We get candidate gene list, and then you try to validate and do the science. So this is a 10-year, at least 10-year process. The project that I introduced to you are actually 20-year projects. So this is really too long. Maybe with GWAS, now we can be faster, and as I said, if we have, we can do this in the computer. I mean, this is a dream come true for me. So I think breeders become interested at this point, not so much before, so they want something validated, breeding ready. Again, a problem for researchers to get a project to that stage, that breeders really take it on, because they want to have a large effect, 5 to 10%, no yield penalty under control, as I said, effective in different genetic backgrounds, in different environments, and you name it. So really, there's a need to link up with breeding programs at an early stage to make sure this is getting, that they help us with that. I think industry then is more at the other end of the spectrum when they become interested in candidate gene list. But again, also there, major validated genes is what they love. And then they put it into their pipeline, test it as GM trait or as hybrid, as a delivery mechanism. There, the minimum requirement is 10% yield advantage under on average, because otherwise the investment is not really worth it. Again, effective in different genetic backgrounds and different environments. So this is a lot of things to do, from gene discovery to get it out there, and it's really something that anyone alone or any small research or universities really can't do. So we need to think about how we can better do this and have strategic partnerships to get this done. This is my last slide. This is a photo I took from our sub-1 breeders, or farmers actually, in India. So he was pretty happy and pleased with what he got there. And I think really the point I want to make is if we want to have projects like this and success and make a difference, we need more longer term funding. We need to tap into this genetic diversity, and there are global efforts going on in wheat and rice as well to develop populations to get access to this gene pool. We need links with breeders and industry to help us get this done. And also, I would probably think if there were more researchers now moving from our Arabidopsis into relevant genotypes, that would help as well in the gene discovery world. And we need also to be specific about strategic about the stresses we address. And I think drought, heat, P fixation, rather than P deficiency, but P fixation is a bigger issue, I think, and internal nutrient use efficiency. So enhancing internal nutrient use efficiencies to have plants that dilute without a yield penalty, be able to produce more with really less nutrients is, I think, it's the way forward, especially for resource-poor countries. And this is my last slide. Thank you very much.
Thank you very much for an excellent paper. Questions for Dr. Hoyer? Chair's prerogative on the first one. The phosphorus work in rice, is it a greater uptake of phosphorus, or does the gene actually allow more efficient use of phosphorus?
It's more uptake. It's just because the root surface area is larger, because it's growing better. It's better uptake. Yeah, so we have done 2 microarray studies to look into the starvation or the genes responsive, and it's usually not the starvation genes that we found. It's more the root growth and drought responsive genes.
Thank you.
Yeah.
Other questions from the audience? Yes, please.
So, for the Qtl study on the drought tolerance, how confident you are like Qtl is stable? Do you have like a multi-environment or multiple testing about the different environment, the stability of those Qtls for the drought tolerance?
Yes.
You are showing how much you are confident about this Qtl is stable in the wheat.
When we consider it stable.
So you have to talk about the genes for the drought tolerance. So do you have a test or multiple environment which is proving that this is a stable?
Yeah, so the graph I showed, this is actually, I think, 4 different sites. We also work with CIMMYT to test those materials in Obregon. And really the outcome of this testing was that it's a matter of deeper soil or shallow soil that is really determining if the alleles or the Qtls are effective or not.
All right.
And
Thanks.
yeah.
Let's thank Dr. Hoyer for an excellent talk. Our next presenter is Dr. Zaire Khan. You heard a little bit of pre-discussion this morning from John Pickett. Dr. Khan is the Thomas Risley Odhiambo Distinguished Fellow and Principal Scientist at ICIPE, the International Center for Insect Physiology and Ecology. Dr. Khan has had a remarkable career. I would just point out that in 2015, he won the Louis Malali Prize for Outstanding Career in Agriculture, awarded by the Agropolis Foundation. So congratulations, and the podium is yours. Thank you.
Thank you very much. Thank Maurice for inviting me and giving an introduction on the first day about the push-pull program. How do you change?
Big green is,
the big green one, okay. Without taking a lot of time, this is a work which I started 23 years ago at International Center of Insect Physiology and Ecology in collaboration with John Pickett of Rothamsted, who talked a little bit about push-pull this morning. We have a website which is www.pushpull.net. So most of our publications and brochures for the farmers and everything can be downloaded from there. And the history of the push-pull and how it works, it is well described there. Coming to cereals, the push-pull which we developed is for cereals. The cereals in Africa face 2 major problems: the stem borers and striga weed. Stem borers are moths which lay their egg on the cereals. The larvae, they feed on the leaf and they penetrate inside the stem and feed inside and damaging the vascular system. Striga is a parasitic plant which produces the pink flower. Each plant produces about 10,000 seed. They remain in the soil viable until the next planting season, or they can remain viable for at least 8 to 10 years if you don't grow a cereal in next year or third year or fourth year. Both of these pests completely are devastating. They cause the total loss to the cereal. The annual loss to the cereal in sub-Saharan Africa is about $7 billion. That's the annual loss. and these effects are aggravated by degraded soil and climate change. We are seeing that Striga is moving to new places where it was not there because of the drought situations. What is push-pull? Push-pull is a novel system which was thought by us, and it is developed by understanding the complex mechanisms that govern the ecology of plants and insects. It uses two kinds of plants. We use two kinds of plants on the border. We use a trap plant, and in between the cereal we plant a repellent plant. The repellent, both of these plants are perennial, and we selected in a way that both these plants could be useful to the farmers. Both these plants are fodder for the cattle, and then the intercrop plant with the cereal, it produces the volatile chemical which repels the stem borers. The stem borers are attracted by the border plant, which is highly attractive, about 5 times more attractive than the cereal crop which is planted, and but it does not allow the development of the insect. The intercrop also recruits the natural enemies. We selected the intercrop which was highly attractive to natural enemies. So whatever the larvae or eggs are left in the field, they are killed by the natural enemies. There are various benefits, although we developed the push-pull initially only for the stem borer control, but later we kept on modifying it and developing it, and at present it controls the parasitic Striga weed. The both push and pull plant, they provide fodder and helpful in milk production. It improves soil fertility. We are doing a lot of work on the soil. Climate change adaptation, we adapted it to climate change, and also it mitigates the climate change. We learned the lesson from the grasses. When I came to Africa, I looked at the grasses, and I found that the grasses and maize are growing at the same place, and they share the same stem borers, but the maize is heavily infested by the stem borer, but the grasses know how to defend themselves. So that was the start of this work, to understand how the grasses can defend themselves while the maize is heavily infested by the stem borer. What are the characteristics, and how? And we found later that the grasses have developed the strategies which are very smart, and they defend themselves as well as they can defend the neighboring plants against the pest attack. And from there we have started looking into the grasses, and surveyed about 600 species of grasses, which took us 2 years to go and survey and look for their association with different stem borers, and how these stem borers how these stem borers affect the grasses, and how the grasses affect the stem borers. We selected a grass which is known as Nipia grass. It is very well known fodder, and first time we found that the Nipia grass was 5 times more attractive to a stem borer egg laying. If you give a choice to the stem borer to lay the egg in between the maize plant and Nipia grass, 5 times more eggs could be laid on the Nipia grass. This was the first discovery we had, but very few of them could become the adult. If you look at the graph, about 70% of those eggs which are laid on the maize could become adult, whereas on the Nipia grass, only 5 to 10% can become adult. Whereas when you look at the other comparison of the graph, there are 5 times more egg laying on the several varieties of the Nipia grass. And this was something very interesting, and we said, can we use it? Nipia grass is a good fodder plant. Can it be used as a trap plant to control the stem borer? And it will reduce the pesticide use, heavily pesticide use. The question was why the Nipia grass is so attractive. And with John Pickett's lab, we started looking into the volatile production by Nipia grass and maize plant, and compared it into different hours of day and night. And we found that as soon as it becomes dark in the first phase of escotophase, the Nipia grass starts producing 5 times more the attractive chemicals like hexanol and hexanyl acetate as compared to the maize plant. And that is the time when the stem borers are also looking for the host, and they find the Nipia grass highly attractive, and they lay their egg more than they lay their egg on the maize plant. So if the maize plant is growing next to the Nipia grass, almost 5 times eggs will be laid on the Nipia grass because of this phenomenon, which this plant can do, which maize cannot do. Later we found that this volatile production cycle is also present in several other grasses, and that is what the nature has provided to these grasses to defend themselves. Because they are under continuous attack by the stem borers, and they can recruit the stem borers to come and lay egg, but don't allow the stem borer to develop themselves, and that's how they have co-evolved and co-survived with the stem borers. The other very interesting plant we found was during our survey was Melanis mintiflora. This is another the other grass, African grass, and it has a very interesting phenomenon that it repels the stem borer. It highly repellent to the stem borer. A stem borer will not lay egg on the Melanis mintiflora, and if Melanis mintiflora is intercropped with the maize, even after 10 rows of maize, 1 row of Melanis mintiflora, it will significantly reduce the stem borer egg laying. But it attracts the natural enemy to in the field. So whatever eggs are laid, the natural enemies can come and kill those stem borer eggs. And this was the only grass which we could find which could do this. It can attract the natural enemies without being damaged in the field, and it was such a novel finding that we published in Nature in 1997. And we found that it produces some very novel chemicals like osamine, dimethyl nonatrine, caryophyllene, and other chemicals which are repellent to the stem borer but attractive to the natural enemies. Recently, we are finding that these molasses grass can give signal to the maize plant. If maize plant is growing next to the molasses grass, the maize plant learns from the molasses grass also to produce the same chemicals, and it starts attracting the natural enemies. So in our lab, we are now looking into the mechanism of the production of these chemicals and how the molasses grass can induce such defense signals to maize plant. So this is some of the work which we are doing. Right in 1996, after discovering that the Nipia grass could attract the stem borer and molasses grass could repel, we thought that we should involve the farmers. It took us some time to learn and bring some social scientists and talk to the farmers, and farmers have never heard that the grasses could be used to control the stem borers. But we tried to show them how does it work in our greenhouses and how does it work in our experiment, and they believed it, and they said, yes, we could do that, we could participate in your study, but we would like to make sure that these grasses are of economic use. So if you bring us something which our cattle can feed, and fortunately both these plants we had selected were good fodder plants, so the farmers adopted it, and we showed them in the field that how in this kind of arrangement where molasses grass was planted in between maize plant and Nipia grass was planted in the border, could give a 60% reduction in the stem borer control, which was very encouraging for the farmers. But this work we did in the area where striga was not present. This was our first experiment where there was no striga. But when we went to the area where there striga was present, the farmers opposed the technology. They said that why should we control a stem borer when astraga is so devastating? So can you find a technology which could also control the astraga? And we being entomologists, we had never worked on astraga. But fortunately, we had a discovery. It was very accidental discovery. We found a legume. When we were looking for a legume which could repel the stem borer, we found that Desmodium uncinnatum not only could repel the stem borer, but was also able to control astraga weed. And that was a breakthrough. We went back again to the farmers which were not adopting the technology in astraga controlled area and showed them how just intercropping the Desmodium with maize plant could control the astraga. As you see on the right-hand side, it was intercropped with the maize is so healthy, whereas on the left-hand side, the astraga has killed all the maize plant and the stem borer has killed the maize plant. The farmers were very excited. We planted other legumes like beans, cowpea, green gram, maize and Desmodium, and showed that Desmodium was superior in controlling the astraga and the stem borer. The farmers adopted it, and later we did some chemistry, and still doing the chemistry, we found several flavones and isoflavones which have two different kind of chemicals. The one group of chemicals, it stimulates the astraga seed to germinate, but the other group of chemical does not allow its attachment with the maize plant, and that's how the astraga seed bank can reduce significantly when the maize plant is intercropped with Desmodium. The farmers adopted it very fast. You can see the before and after, how it has changed the life of the farmer. The farmer which was not growing anything was losing the crop year after year, can grow. And this is a farmer which is disabled farmer in Western Kenya, and she adopted the technology after a lot of thinking because she was not able to work in her farm because of the astraga and was not getting everything. And now for last 3 years, she is getting enough not only to feed herself and family, but also to sell enough maize. As you see here, we know the chemistry very well now. We know the attractive and the chemistry which attracts and repels the stem borer, the root chemistry, and the system works, the push-pull system. It exploits the science and benefits the farmer so well. The farmers have enough fodder from the both push and pull plant. Both, as you know, both these plants are perennial, and once planted, the farmer can take advantage for many years. Some of our farmers have planted it 18 years ago, and they still have push-pull there in their farm, and they have high yield, and they have the fodder, and they are able to have now the cattle, dairy goats, and dairy cows. The fodder production has increased about 3 times, and milk production more than 2 times in average push-pull farmer's field. These are reduction in the stem borer, astraga weed, and increase in the milk, increase in the maize yield. We did also very rigorous economics with a PhD student to show that the push-pull gives return to the labor and also the return to the land. It's very well done. It is much better than the planting the intercropping with the bean and maize monocrop. And we are still continuing with some of the more economics work and farmers' perception work. We have started a new collaboration with Laurie Drinkwater of Cornell University, and she has taken her sabbatic. She was very keen on the push-pull to see that what happens in the long-term push-pull trials, both on the station and in the farmer's field, and she has shown that there's more nitrogen, soil phosphorus, and carbon sequestration in the fields which are planted under the push-pull in the long term. She is also looking into in the farmers' field trials, and the farmers who had planted 18 years ago and the farmers who had planted about 2 years ago, and you can see how the carbon in the soil has increased from the farmers who have recently planted to the farmers who have planted 18 years ago. So that work is going to be very important to us to see that how the push-pull technology can improve the soil health. About 5 years ago, or 6 years ago, the farmers complained that our conventional push-pull is not surviving the long droughts because of the climate change. So we approached the European Union, got new funding to select the new plants, new push plant and new pull plants, which could survive the longer drought, but can give a good protection to the crop. So we have a new push-pull technology now, which we call climate smart push-pull. It uses bracharia and uses desmodium in totem, which is green leaf desmodium, and it can work with both maize and sorghum. And it does the same thing which conventional push-pull does, but it has other benefits than the conventional push-pull. We have also discovered during our survey, we have also discovered some new desmodium which are of African origin, and these are highly drought tolerant. They could grow without a drop of water for almost 6 months and can survive without water for several more months, and they can regenerate after the rainfall. So this will be our future desmodium which we'll be using in our field. And we are doing the field trials, as you see here, with and without. On the right hand side, you have sorghum with desmodium, and left hand side without desmodium. And it also has some very novel chemistry which we are continuing to work, novel chemistry, and publishing some of this work, which is of quite interest. We are looking into the gender preference, and you see that we have two kind of push-pull, a conventional push-pull which is in dark blue, and climate smart push-pull is in a gray color in the graph. About more than 122,000 farmers, and now today is more than 125,000 farmers. Most of them are recorded by GPS. They have adopted the push-pull. And now, as you can see there, very few farmers are adopting, new farmers are adopting the conventional push-pull. Most of the farmers are going for climate smart push-pull since we have discovered it in 2012. The women especially like the conventional, they like the climate smart push-pull because it uses the bracharia. It is much easier for them to harvest and handle and feed to the cattle, as compared to Nipia grass, which was very hard, and they had always to wait for the men to come and harvest and help them. So many of the families are female-headed, and they like the bracharia, and they prefer the climate smart push-pull. As you see here, the adoption in the green color is by women farmers, and the more and more women farmers are adopting the climate smart push-pull as compared to the men. And that is also a success for us. We are also discovering some new science, and some of the new science is based on early herbivory alert and plant signaling, and this is going to make our push-pull much smarter. We discovered that bracharia brachyantha, which is one of the parent of the bracharia we are using, is a smart grass, and it has early herbivory alert. Early herbivory alert means as soon as eggs are laid, even before the larvae emerge and start feeding on the grass starts producing the chemicals to defend itself and starts attracting the natural enemies. But what bracharia brachyantha does, that is, it stops producing the chemical which are attractive to the stem borer. So no further egg laying on this plant, but starts producing the chemical which are attractive to the natural enemies. And those natural enemies come and wait for the larva to emerge from the egg, and it attacks on the larva. So these are very intelligent and smart traits which are present in nature has provided to some of these grasses, which we need to understand and probably bring it to our maize plant so that the maize could be also as smart as the grasses are. And that was a question which we raised. But later we found that, later we found that the maize can also do that, but not the hybrid maize, because during the breeding, these traits have been lost. But the land races and the OPVs, which are with the farmers, they can produce the early herbivory alert. Not only can produce, but also signal it to the next plant, maize plant, if the attack has started. So we are working on it, trying to look into the genetics of it and identifying the gene so that these genes could be brought to the hybrid plant, so the hybrid plant could also be able to defend itself. So all this new science could be incorporated into our new push-pull program. What is the scope of the push-pull? There are more than 300 million smallholder farmers in sub-Saharan Africa which suffer from stem borer and striga, and this technology can be taken to those farmers. So we are now moving from the Eastern Africa to the Southern Africa and Western Africa so that more and more farmers can adopt. We are working with many NGOs like One Acre Fund, which already has about 300,000 farmers, which they are going to benefit from the push-pull because we are working with them. And although it was originally developed for maize, it can be equally applied for sorghum, millet, teff, and upland rice. We have shown that it can work with all these crops. But also it can be adapted, and there's a lot of interest with other crops like cotton, coffee, and other horticultural crops. The push-pull system can be applied and the concept can be applied. We are using various dissemination pathways. Ruth Onyango talked about the diversity in Africa, and we also found that no single pathway can work in disseminating any technology to the knowledge-intensive technology to the farmers. So we are testing various dissemination pathways, and we are testing and trying to show that which pathway is better for which community, so we can spend the donors' dollars on those pathways to go to these. And this is work which we are doing with our PhD and postdoctoral students to see that how the farmers could adopt the knowledge-intensive technologies and which pathways are the best for which community. The push-pull has been mentioned by UN General Assembly report. Recently, the Secretary General has mentioned it in the UN as a technology which will help to increase food production in Africa and beyond. And there's a website to it. And we believe that the push-pull is the kind of technology which is necessary to support the green revolution in Africa, because it increases productivity without requiring extra resources. The farmers in Africa are poor. They don't have extra resources. They don't have money for fertilizer and hybrid seed. Many of them are struggling whether to pay the school fee or to buy the fertilizer or to buy the seed. And if we are able to double the yield without buying the hybrid seed or applying the fertilizer, that will be great for Africa. This is one way I see. And our recent developments in the push-pull on plant signaling, soil health improvement, climate change mitigation, and drought resilience, it represents the greater opportunity for further enhancing the effectiveness of the push-pull. And we see that the push-pull could be adopted not only in Africa, but beyond Africa. Thank you very much.
Thank you very much. Questions for Dr. Khan? Rob Sagan.
In your research work that you've done with push-pull, are there any parts of the production system that are sacrificed in the process? And my question is there any parts of the weed control process that are sacrificed? If we're starting with a soil that is basically drained of fertility, there is no zinc, there is no, copper, there is no potassium, how does a farmer, implement push-pull and at the same time try to build up soil when there's nothing coming in? And I just genuinely don't understand, so I'm asking the question.
Yes, the farmers, if they don't have very poor soil, the desmodium being perennial, it slowly improves the soil. The farmer use to feed the Nipia and desmodium to the cattle and bring back the manure to the field. So we show them the process of bringing back the manure to the field to build the soil, and the farmers do that.
Yeah, I had a question about the adoption of push-pull.
Yes.
And I think you said in some areas of Africa, it's not as popular as others. Is that just extension work, education work, or why did you say that?
No, I did not say that. I said that there are different communities and they learn in different ways. Some communities like to learn from early adopters. Some communities like to learn through the radio. Some communities try to learn through the farmer teachers. So we try to understand that how they would like to learn about a technology and use that method to introduce the technology to that community. That's what I told. Because of the diversity of.
Last question, Morris.
So as you've looked across the application of push-pull, Zaire, could you give us a sense as to the range of improvements that you are seeing in the maize crop, for example? From what to what do you think is what you're seeing across various regions?
The yield of the maize could be less than a ton to about 3.5 ton. It is average. Some farmers could get even more than that. It comes in maybe in 2 to 3 seasons of continued doing that, because the soil has to build, the fertility has to build up. The milk, after the farmers gets the dairy cow or dairy goat, there's milk in the household. There's a better nutrition. We have started some new work on the nutrition to see the nutrition. We just finished our first survey. So we see various improvements in the livelihood of the farmers. I brought a book which was our success story, which we asked the farmers about how it has increased their livelihoods, and we compiled that information to look into the improvement in their livelihoods.
Let's thank Dr. Khan for having us. Thank you very much. Our final session this afternoon is Dr. Tim Sharble. Tim is the research chair in seed biology for the Global Institute of Food Security right here in Saskatoon. He's had a significant research career working at McGill University in Montreal, time at the Max Planck Institute, and today he's going to talk to us about one of his specialties, Apomixis. Welcome.
So I'd like to thank the organizers for inviting me to give a talk here. And before I begin my talk, I'd like to thank my group. I'm in the process of moving my research group from Germany to GIFS, and these are the people that do all of the hard work and generate all of the cool data, and so I'd like to start by thanking them. So I like to talk about sex, and I thought I would start off with a 2-minute short animal porno flick. And so, I'm showing you this film for a point. So I promise you'll understand why I'm showing you this. So the context of this is that these are about 1 centimeter long flatworms that live on the Great Barrier Reef. They're hermaphrodites, but they can't self-fertilize themselves. So you have to fertilize another individual, and the way they do it is that their penises have evolved into little syringes, and these syringes can be used to inject sperm anywhere in the body of your partner. Okay, so that's how you reproduce. So let's do the film. It's just 2 minutes. Do I start it, or can you guys start it? It's a National Geographic film, so we can just watch it.
Flatworms have both male and female sex organs. But when it comes time to make little flatworms, they have to decide who plays which role. And that they fight over.
There you can see the penises
The two
coming up there.
flatworms rear up, exposing their midsections. Those jutting white nibs aren't weapons, at least not intentional ones. They are actually the worm's penises, a double-barreled inseminator, if they can shoot first. Flatworm sex consists of the two attempting to stab their lover with their pointed pair and inseminate each other, an act delicately referred to as penis fencing. But there's nothing delicate about it. Flatworms have been known to gouge holes in each other in battles lasting up to an hour. Why do they cross swords? Producing offspring requires a lot of energy. The winning father can go out and about in the world without any further responsibility. The losing mother absorbs the winner's sperm. It must now work longer and harder to find food and stay alive. The father floats merrily along. The mother crawls along looking for a meal, already the responsible one.
So not too far off from humans. I guess so why did I tell you that? So why I told you that is, I mean, this is an extreme example of sexual selection. And the point I'm just trying, and you can see from these pictures, is that these things actually rip each other to shreds when they're reproducing. So, you know, and the point I'm just trying to make is that natural selection for sexual reproduction is an incredibly strong force. And when you go to look at plants, for example, so on the left-hand side, this is just, we all know what sex is in plants. It's very well defined, very highly constrained, and very difficult to find variability upon which we can act upon, right? And in apomictic plants, so I'm going to talk to you a little bit about why Apomixis is such an important technology for us to undertake. But Apomixis, so on the right-hand side of that diagram, you can see that rather than having genetic material coming from the mother and the father, the female actually makes a meiotically unreduced egg cell, which develops into a genetic clone of herself. And males actually have very little to do with reproduction. And so the other point, besides the goal of putting Apomixis into plants, what I would like to say is that Apomixis has a wealth of information in it. They're natural mutational laboratories that enable us to look at pollen formation, egg cell formation, endosperm formation, and so on and so on. So that's just my little thing. All right, so but I'm standing here because, how can refraining from sex help humanity? So I think we've all seen this, and people have alluded to this today, right? So many of our crops depend on hybrid technology, and hybrid technology and exploiting heterosis leads to yield increase. Now, in things like, in pulses where you have cleistogamy, it's very, we haven't been able to exploit heterosis because crossing schemes are very difficult in plants that fertilize the closed flowers. So, so the reason we're studying Apomixis is because Apomixis is a tool that could lead to a fixation of heterozygosity and hopefully heterosis in crop plants. And so this is just, we know what this is, a typical breeding scheme. The thing is, if a farmer takes those, grows the F1 plants, right? They get the seeds from the companies, they grow the plants up, the plants are all genetically uniform, but then that farmer takes those plants and crosses them again, then sex and meiosis just mixes everything up. So you lose your traits, you have increased variability in the next generation and so on. So, what it means for the farmer is the farmer, of course, buys seeds every year. If the idea that we're working on is that if you could take that F1 population and then simply turn sex off and turn on Apomixis, then those plants would reproduce clonally from then on. And while that, it sounds really bad for companies, it's not real. I told you that last statement about buying seeds one time and never buying them again. That's what most Apomixis researchers have been saying this for years, right? And so I don't believe that, I'm just stating it. But nonetheless, when people talk about the introduction of Apomixis technology into crops, they tend to use the word billions. So it would change things. It's a very disruptive technology. So on the left, if we think about hybrid crops now, we have a significant investment required, time and resources to create, to recreate these F1 hybrids every generation. And the idea would be that if we could introduce Apomixis, you could make single generation hybrids, right? So you could cross plant X with some distant germplasm, let meiosis and sex mix things up for you, right? Throw the seeds out into some selective regime, and that could be temperature or light or whatever you want. You choose the best phenotype that grows out of that in that selective regime, turn off sex, and go to seed increase right away. So that and so the idea would be that we could both quicken the pace of the generation of new hybrid plants, and secondly, we could utilize a lot more of the biodiversity in the world. So I think really when I think about Apomixis, I think about diversification. So we think about single generation hybrids. We could potentially produce unlimited numbers of hybrid crops. It brings up the concept of niche breeding, and furthermore, I think farmers, local farmers who have locally adapted types of crops those farmers could actually be brought into breeding programs, because we could very rapidly breed or exploit some of these traits using Apomixis technology. So there's local and global benefits. So my research group, how much time am I doing here? You're doing well. You have about 15 minutes. Okay, so in my group, we basically approach Apomixis, understanding Apomixis. We look at naturally occurring apomictic plants, and our goal is to really utilize a lot of the literature in human disease research. And so what we endeavor to do is to understand these natural populations of asexual plants or apomictic plants. We very well define the phenotype, and we're really getting down to cell-specific analyses, right? So we're interested in pollen formation and egg cell formation and so on and so on. I work on a number of species. The one I'm going to talk to you about today is one I've been working on for almost 20 years. So the genus is Buchera, or Buchera. It's a crucifer. It's related to Arabidopsis thaliana. A lot of genome synteny there. It's closely related to Brassica. And the idea would be that if we can identify genes for Apomixis in Buchera, then we could apply them relatively rapidly to Brassica. So let's just talk quickly about phenotypic variability. So an apomictic plant it's not an on and off, it's not a 0-1 trait. Very often apomictic plants are facultative, so they produce a certain proportion of their seeds apomictically, which means that when you're analyzing the phenotype, you have to sample many seeds from a single plant to understand what the penetrance of the trait is. And so we do this using flow cytometry. And so this just shows you normal sexual reproduction. We have double fertilization in angiosperms. So one sperm cell is used to fertilize the egg cell. One sperm cell is used to fertilize the binucleate central cell. And you end up with an embryo that is diploid and an endosperm that is triploid. That, and furthermore, endosperm formation, you have 2 maternal genomes and 1 paternal genome. There's tons of genomic imprinting involved in that process. And typically, when you tweak any of those genes, it leads to seed abortion. So we don't learn anything from these plants. Now, interestingly, an apomictic plant, in the case of Buchera here, can produce, on the left side, it can produce certain levels of meiotically reduced pollen and meiotically unreduced pollen. And furthermore, the same plant can produce meiotically reduced egg sacs or ovules, and meiotically reduced and unreduced. By definition, there's no fertilization of the egg cell, as you can see with the big X's in the bottom, but very often we have fertilization of the central cell. And we could talk about that, but I'm not going to go into details here because of my time. The point being that when you look at the seeds of an apomictic plant, you're looking for deviations from the 2-to-3 genome ratio of the embryo and the endosperm. And furthermore, you're doing this by looking at hundreds of seeds from a single plant. So a couple of years, or 2007, one of my postdocs, Wally, we developed a high-throughput flow cytometric method. This is a paper we published in 2010, and it's simply, on the y-axis shows you penetrance, so percentage of seeds that are produced apomictically, 1 being 100%, 0 being sexual, or 0%. And every dot represents the average of 3 genotypes, 3 individuals of a genotype. And I've ranked them from low to high. And what you can see is that we have obligate sexual things at the lower left. We have obligate apomictic things at the upper right. Circles are diploids, squares are triploids. And furthermore, we have a little bit of leakiness, so facultative apomixis, so we have things that produce 5 to 10% apomictic seed at the bottom, and things that are producing 80 or 90% apomictic seed at the top. Why is this important? It's important because apomixis, it starts in a developing ovule, right? So when we're and I'm going to show you in a minute, when we're actually doing our omics analyses of these developing ovules, you have no idea when you're microdissecting a cell whether that egg cell is going to be apomictic or sexual. So very importantly, having this kind of data enables us to choose things that are 100% apomictic and compare these things to 0% apomictic. So we know we're actually comparing those two traits, right, without any mixture of anything that's not reproducing apomictically. Quick time? How much time?
Do not be alarmed, but 10 minutes.
10, perfect. No, we're right on time. Okay, so we do these analyses, right? Oh yeah, the last thing I want to say is that graph I showed you, right? So my postdoc, Wally, he analyzed 22,000 single seeds. This took months. It's unprecedented. People have not been able to do this in the past, right? So it's an incredibly informative analysis that enables us to really understand, to try to connect these thousands, if not millions, of polymorphic markers with penetrance of the trait. It's a very, very important thing to do. All right, when you do those analyses, what we discover is that 95% of the diploid apomictic bucera that we've analyzed produce seeds with a diploid embryo and a hexaploid endosperm. Now, why is that? Well, it tells us two things, potentially tells us two things. It tells us that these things are producing meiotically unreduced egg cells and sperm cells, and for some reason, there seems to be selection for this hexaploid endosperm. Now, you recall when we talk about sexual endosperm, we have 2 maternal genomes and 1 paternal genome to make them triploid. In an apomictic plant that has a hexaploid endosperm, we have 4 maternal genomes and 2 paternal genomes. So we don't disrupt the ratio. So we think this is one of the reasons why there's a maintenance for the fertilization of the central cell. So the hypothesis is meiotically unreduced egg cells, meiotically reduced pollen cells. This is the first step in what we think is in the pathway to apomictic seed development. So we decided to have 2 projects. One was led by one of my postdocs, Jose. We're interested in the female part of the trait, and what we do is we don't do laser capture or anything. We do live microdissection. We're trying to minimize degradation of mRNAs and things. And what we can do is, on a good day without not too much coffee, you can go in and you can actually micropipette, like 10 or 20 ovules, and we can get a good, transcriptomic analysis of that. So we did that a couple of, this was done like in 2009 or something like that. And we go to microarrays. We're not using NGS at this time. I can talk to you about that later if anyone's interested. But the important thing is that we're comparing multiple genotypes of sexual individuals and apomictic individuals. Now, if you compare any 2 things, they're going to be genetically related to a certain extent, but you're always going to find differences between them. So by having lots of genetic backgrounds of sexuals, lots of genetic backgrounds of apomix, what you're doing is statistically looking for things that are the same in all sexuals, but different, and yet the same in all apomix. And when we did this analysis, corrections for multiple tests, and so on, we find 4 probes on our array, and lo and behold, those 4 probes nail 1 gene. So it's like technical replication, biological replication. Everything points to this gene as an important gene. My postdoc called it Apollo. I didn't. I don't take responsibility for that. But to make a long story short, if you do the sequencing, sexual plants have sexual alleles, and apomictic plants always have a sexual allele and an apomictic allele. And the apomictic allele differs by a functional polymorphism in the 5' UTR. Importantly, if you look in somatic cells, so roots, leaves, and so on, the gene Apollo is always expressed. So it's a housekeeping. It's associated with the cell cycle. And more importantly, in sexual plants, Apollo is turned off, whereas in apomictic plants, it's turned on by expression of the apomictic allele. So. And we published this a few years ago. Now, we're interested in evolutionary biology and population genetics. Conservation means importance, and in red, so we've looked at apomictic bucera from all over North America. Blue is sexual, red is apomictic, and red demonstrates the conservation of this apomixis allele across North America. So it's highly conserved, and therefore we hypothesize that it's very important. All right, that's the female story. Talk about the male story. This is done by a PhD student at the time in my group, who's now a postdoc in Germany in my group, and we were looking at anther head formation. And so Martin spent a lot of time trying to identify at which point do do sexual and apomictic anthers differ with respect to meiotically reduced or meiotically unreduced egg cells? We went back to our microarrays, same kind of experiment, completely different tissues and so on. This time around, we nailed 3 probes on the array, and those 3 probes hit 1 gene. So now, is it the same gene or is it a different gene? It's a different gene. So Martin called this thing upgrade, unreduced pollen grain development, and I'm not even going to begin trying to explain this diagram to you. The take-home message, though, is that this is essentially the first Apomixis-specific gene ever identified in plants. This thing, it's a chimera between at least 3 other genes that we've been able to identify. And furthermore, the original genes to which this chimera corresponds are found in both sexual and apomictic plants. So we think that this gene is post-transcriptionally regulating other genes in the genome, and it's very difficult to explain its origin because of this chimeric structure. And finally, most importantly, as with Apollo gene, this upgrade gene is highly conserved across North America. Now, you recall at the beginning of this whole this part of my talk, I said that most apomictic plants make a diploid embryo and a hexaploid endosperm. Therefore, we need meiotically unreduced egg cells and pollen cells. I've shown you 2 different genes which are highly correlated with the production of those 2 things. Therefore, if our hypothesis is to be supported, we would expect both these genes to be found together. And lo and behold, if you look in black across North America, these are all individual plants that are apomictic that share both of those genes. So once again, we think that this is highly suggestive that the genes we have are the genes, in fact, that are underlying this trait. So that's pretty much it. I only had half an hour, so I wanted to stop. So what I think is, clearly there's a lot more to the story we could talk about, right? So I was just being quick and showing you some of the highlights. But we really do think we have our foot in the door. And so I've shown you that there's a separate mechanism for, apomictic or apomeiotic egg cell and pollen cell formation. There apparently is a different factor for both of these things. And what we're looking at right now is to try to figure out where they came from. And what our impression is that these things have been independently derived during the evolution of Bucera, kind of in a post-Pleistocene glaciation context of North America. What we are doing, though, is we have some, besides my funding from GIFs, besides my funding from Germany, I also have some venture capital funding. And what we've just received a lot of seeds from Arabidopsis and maize, and we're awaiting brassica right now with these codon-optimized constructs containing Apollo. And what we're trying to do right now is induce the trait or anything that resembles that trait in these plants. And with that, I think I'll say thank you for listening. Give you a little bit of Darwin.
Questions for Dr. Sharble? Yes. If you shout it, maybe we can repeat it as well.
Thank you. So I have a flippant comment and then a question. So my flippant comment is, of course, Apollo should have been Aphrodite.
Good point. But it didn't match.
And secondly, if you look at the evolution of land plants, and you look back on all the genomes of the very primitive of the first of the plants to make it out of the primeval soup, how ancient is Upgrade and how ancient is Aphrodite?
So Upgrade only exists in apomictic Bucera. It's not found in any other plant, first of all. Okay. Apollo.
So it's not found in mosses?
Upgrade is a chimera. So it was literally. This occurred in Bucera. So sometime during the cladogenesis of Bucera, and we're looking at this now, right? It's not. So if you go, Bucera, and then there's Buceraceae. There's things outside the phylogeny, and if you start going it back, Upgrade is not there. It's only in apomictic Bucera. So it's really recently derived.
So is it by horizontal gene transfer?
No, it's as I said, it's a chimera. So it's some sort of genesis. I can't answer that. I don't know what it is, and we're trying to determine this. Now, so Aphrodite, this gene is found, in the plant kingdom, and so it's in fact there's orthologs even in animals if you go, if you relax homology constraints and things like that. So it's a gene associated with mitosis, and it's not the gene itself which is important, it's the functional polymorphism which is important. And without going into all the details about it, we're unclear, or what we're looking at is whether that functional polymorphism both introduces novel transcription factor binding sites into the genome, and it also removes some. And so what we're presently also looking into is the titer of these transcription factors. And we're thinking that maybe Apollo acts as a sink either as a sink or, or something along those lines. I'm not sure though. So does that answer your question?
Sort of, but it's great stuff. Thank
Yeah.
you.
Other questions? Yes, at the mic.
Tim, what puzzles me is that, well, I've never heard about Bucra, but you have all this genetic information from so many locations in North America. Why is that?
Why do I have so much information?
Well,
I mean, great collaborators.
This plant is not very well known.
Oh, no, it's not. So in 1999, I joined the Max Planck Institute for Chemical Ecology in Jena, in Germany, and my supervisor at the time was Tom Mitchell-Olds, who was a visionary. And when I joined his group and I told him I wanted to work on asexuality, I was in his office, he turned around and said, "Here," and gave me 2 big boxes of seed material that he'd collected all over the place. And that was the beginning. And the Bucra community, it's a relatively small community, but we're very tight. We're all very good friends, and we exchange material like crazy. So that's where I have all of that stuff.
Chair's prerogative. Working with those next set of various crops, what increases the candidates that you might want to use for those cassettes that you're working on or other things?
I don't understand the question.
What's the next, so you said that you want to introduce this into various crops.
Yes, yes.
What are those priority
Oh.
characteristics of those crops that will make this work?
We'll see. So it's a little bit of a fishing experiment now, right? So we're looking just for some quick and dirty information that shows that it's doing something. I mean, meanwhile, I've got these other projects going along, right? But I think the evidence is compelling enough, or it is compelling enough, really, to say, okay, let's go fishing and just try it. But I've recently hired a postdoc from China he's my genome editing person, so we're already thinking about that. I will point out there's one final little kind of secret thing. We've discovered what apparently is an interaction between Apollo and Upgrade. So I'll just leave you with that piece of information, which is very interesting.
So given that other species are intermixing, do you think there's a shared mechanism, or could you find something completely different?
Yeah.
Something like dandelion?
No, I think it's an excellent question, and it's probably a different mechanism. Well, no, it's probably different genes. Let me answer that correctly. Every apomictic species that people look at, and so we're looking at about 6 different monocots and dicots in my group, and in each of those cases, so sexuality is the ancestral trait in all of these things. So sexuality, or apomixis, was derived from sexuality repeatedly during the evolution of these different plant systems. Now, apomixis tends to deal with modifications of meiosis. So clearly you only have kind of a certain set of genes to work with, right? So definitely it's conserved in that sense, right? But the mechanism to start the whole process off, I think in a neo-Darwinian fashion, so it's a mutational process, right? So it's probably a different kind of starting gun that kind of generates the process each time. And hence the reason why we want to look at so many different apomictic species, because each of those represents a different toolbox in my perspective.
Let's thank Dr. Sharble.
Thanks.
I've got to say, this is the kind of session that reminds me why I love agricultural science. We are back here at 3:45 for the next section of crop performance and nutritional quality. The standard snacks and reception are out there, or at least the break, and then we're back here at 3:45. Thank you very much for your participation.